vient de l'Institut de la grippe à Saint-Pétersbourg, qui est un des centres de l'OMS, centre de référence de l'OMS pour la Russie, pour tout ce qui concerne la grippe. Son domaine d'excellence concerne l'évolution des souches de grippe en Russie. Quelque chose comme 5 à 600 échantillons par an en provenance de les parties de la Russie et de ce qu'on appelle le CIS, c'est-à-dire des pays qui étaient dans l'Union soviétique avant. Son laboratoire, son institut, est un des instituts associés à un consortium européen qui est animé, qui est piloté à partir de l'unité des virus émergents de la Timone. Et c'est invité à venir donner un séminaire sur des résultats récents qu'il a eu en matière de souche grippale. Donc, Mikhail, si vous voulez commencer, if you want to start. Pour des raisons pratiques, si au moment des questions, vous souhaitez poser des questions en français, c'est possible, il le comprend très bien.
Mesdames, Mesdemoiselles et Messieurs, permettez-moi de vous saluer de la part de l'Institut de recherche de grippe à Saint-Pétersbourg, en Russie. Je ferai volontiers mon exposé en français, mais malheureusement, depuis très longtemps, je n'ai pas pratiqué mon français, donc je passe en anglais. Our institute was established in 1967 after Asian flu pandemic of 57-62, and it is a national WHO center since 1971. The organizer and first director of Institute of Influenza in St. Petersburg was academician Anatoly Smorodintsev, and here in brief I give you some structure of our institute and its competence. We have several departments: Department of Molecular Virology, which group Laboratory of Molecular Virology and Genetic Engineering, Laboratory of Evolutionary Variability of Influenza Viruses, this is the laboratory which I had, and the Laboratory of Functional Proteomics. There is a Department of Prophylaxis and Chemotherapy, of influenza vaccines laboratory, and of molecular antiviral chemotherapy. And the Department of Biotechnology, 2 laboratories, and the Department of Epidemiology, to be short. And what is interesting, we have a clinical department, specialized clinic on viral infections, adult respiratory clinic, and children respiratory clinic, just for clinical trials of vaccines, of antivirals. And the modern topic of research of our institute are presented here. This is the refinement of influenza surveillance in Russia according to the WHO international recommendations. These are the technologies of the design and production of anti-influenza vaccines of new generation. It means that not only classical vaccines, but genetic engineered vaccines produced with viruses manipulated of genetic engineering. These are the studies of the structure and function of natural and synthetic compounds, design of antiviral preparations with the different mechanisms of actions, fundamentals of molecular genetic of viruses and pathogenesis of influenza infection, and finally the strategy of treatment of acute and chronic influenza diseases. Here are several pictures of my laboratory of evolutionary variability. We have a BSL-3 compartment. We also are dealing with small animals as rats and mice. Here are several pictures. Now let's speak a little about the biology of influenza virus. Influenza virus, it is RNA-containing virus, and it has a segmented genomes. negative sense RNA virus. It has error-prone replication, no proofreading mechanisms, and exists as a quasi-species. A consequence of these properties are the high variability of influenza viruses, two ways of variability: antigenic drift, which goes continuously, and antigenic shift, which is estimated as about 3 times per century. Now we'll speak a little bit in details of these properties. So, influenza virus, 8 segments, which are coding of 11 genes. Now they are speaking even of 12 genes, because there are different reading frames in the same segments. And in principle, there are 254 possible combinations of these genes. And the most interesting are 2 superficial proteins, surface proteins, hemagglutinin, which have 16 subtypes, and neuraminidase, 9 subtypes. And if we look on neuraminidase and hemagglutinin, 16 subtypes of hemagglutinin and 9 subtypes of neuraminidase, there are 144 possible combinations. From them observed 86 combinations, and 83 of them in avian populations. This, what is colored blue, these combinations are present in birds, especially in aquatic birds. These birds, aquatic birds, are actually supposed to be the natural reservoir for all influenza viruses. Also, humans are suffering from influenza, but in humans we have observed normally only 3 first subtypes of hemagglutinin, H1, H2, and H3, and 2 subtypes of neuraminidase, N1 and N2. The other animals which are subjected by influenza are pigs, ferrets, which are very good non-human model of influenza, horses, even cats, felines, and even aquatic mammals. But mostly you see the birds who suffer from different types of subtypes of influenza A. Now several words about antigenic drift and shift. Antigenic drift is a gradual mutational process, gradual changes, because we have error-prone reproduction of RNA. So the mutations are generated very often and are not corrected by some mechanisms. These changes lead to the slight changes of antigenic structure, and it goes continuously. What is antigenic shift? This is full alteration of the virus genotype caused by mixing or reassortment of genes in case of simultaneous infection of a cell by 2 different viruses. And it occurs about 3 times per century. And antigenic drift is at the base of epidemics, while the shift is a base for pandemics, because a complete change of the structure gives a new antigenic subtype of virus. So if we look at the studies of influenza virus, here is the main stages of these studies. They begin with sampling the nasopharyngeal swabs of patients, then isolation of viruses, which is an inevitable process until now, virus identification with different antisera, then antigenic analysis, testing of the susceptibility to antivirals, and finally molecular genetic analysis, which gives us the structure, the genetic structure, and where we can observe certain mutations which lead to new antigenic properties. Isolation of influenza viruses is actually done in the MDCK cell culture. This is the cell culture which is recommended by WHO as the optimal system for the isolation of viruses. These cells are the derivative of kidney epithelium of a dog, spaniel, to precise. Normal MDCK monolayer looks like this. And this is after the incubation of 48 or 72 hours with susceptible virus. You see the complete destruction of cellular monolayer. The cells detach the substrate, and here is the typical cytopathic reaction, cytopathogenic action of a virus. So, virus identification and antigenic analysis is based on two reactions. Reaction of hemaggglutinations and inhibition of hemagglutination. Reaction of hemagglutination is a phenomenon based on the capacity of viruses to bind the surface membrane of red blood cells, erythrocytes, and thus to prevent their sedimentation. We observe it in the plates as hemagglutination. And the inhibition of hemagglutination is based on the competition between antibodies and red blood cells for the viral proteins such as hemagglutinine. So virus is interacting with antibodies, and so it cannot no more produce hemagglutination. And this effect is visually seen as inhibition of hemagglutination. And thus we can study the properties of virus, antigenic properties. We can identify different viruses, and we can make a diagnostics with these two reactions. This is a very lucky property which is present in influenza virus, the capacity to hemagglutination. Here, once more, again, I turn to our institute because we have the network of regional basic virological laboratories. 89 laboratories which cover about 2/3 of the territory of Russia in green, shown from far east to extreme west, from the north to the south, and where we receive samples or viral isolates. They are sent to our institute, and we finally study them in detail. And here is presented the diagram of the 5 last epidemic seasons, the distribution of viruses isolated in Russia according to the subtypes. You see the 3 seasons before the 2009 pandemic, we had all 3 subtypes present in the viral population, H1N1 in blue, then H3N2 in green, and B virus in different proportions. Though when the pandemic has outbreak, we had practically monoetiological situation in this epidemic season, 2009-2010. We have 92% of pandemic virus, H1N1 pandemic virus, and only in the end of season we had about 8% of B virus. In